welcome to this talk on coronavirus testing. Now, the newly emerged coronavirus causing the current pandemic is known as SARS-CoV-2, while the disease it causes is known as COVID-19. And there's been a lot of interest in how scientists test for the SARS-CoV-2 virus, and this talk briefly explains the basics. Now, there are two types of tests that can generally be performed, and the first is a PCR test, or more specifically a reverse transcription PCR test. An RT-PCR stands for reverse transcription polymerase chain reaction. Now, the PCR test detects the genetic code of the virus, or in other words, viral nucleic acid, and therefore PCR tests directly test for the presence of the virus. This test can tell whether someone has a current infection or whether someone harbours the virus, even if they are symptomatic. The other test is widely known as an antibody test. And it's important to point out that an antibody test does not directly test for the presence of the SARS-CoV-2 virus. Antibody tests detect whether someone has previously mounted an immune response to the virus. In effect, an antibody test detects the presence of circulating antibodies made in response to the virus. An antibody test therefore gives an indication of whether someone has previously been infected. However, it's very important to point out that current antibody tests don't definitively prove whether someone is immune to SARS-CoV-2. More sophisticated laboratory tests are needed to determine immunity. So how does the RT-PCR test work? Well, firstly, a sample needs to be taken from the patient, and this sample is normally taken from the nose or throat using a swab. A nose or throat swab is taken because this is the part of the body the virus is most likely to be found. A SARS-CoV-2 virus preferentially infects the cells of the upper respiratory tract. That swab sample then needs to be processed in a laboratory, and the nucleic acid derived from the virus isolated. And the genome of SARS-CoV-2 virus is made of RNA rather than DNA, and so the RNA from the biological sample needs to be extracted. This slide illustrates the basic steps of a PCR reaction. Because the quantity of viral RNA is often very small, it needs to be amplified so that it can be readily detected. PCR, or polymerase chain reaction, amplifies DNA, and so the viral RNA first needs to be converted into DNA. This is the reverse transcription step, whereby RNA is converted into a complementary DNA molecule. In order to specifically amplify only the SARS-CoV-2 RNA, primers are carefully designed that are very specific for the genetic code of the coronavirus. The primers act as a specific template so that the DNA that is amplified is specific to the original genetic code of the SARS-CoV-2 virus. Each round of PCR amplification effectively doubles the quantity of DNA. This is known as exponential amplification. At the end of the PCR, there is enough viral-specific DNA to be detected and analysed. Specialised laboratory equipment is required for the PCR test and therefore can only be performed in laboratories with the correct technical expertise. The final analysis then determines whether the virus is present or not in the original biological sample. The reason for taking nose or throat swabs is because SARS-CoV-2 infects the cells lining the upper respiratory tract. These cells are known as epithelial cells, which form a continuous layer of cells that normally act as an important barrier to pathogens. The virus can sometimes infect cells deeper within the actual lung. If the coronavirus does successfully infect the cells of the lower respiratory tract, it can cause pneumonia, and it is this pneumonia which can be life-threatening. So the SARS-CoV-2 virus replicates within epithelial cells within the upper respiratory tract. It essentially hijacks the cell's own molecular machinery to make new copies of itself. Once new virus particles are formed within cells, they can burst out of the infected epithelial cells and spread throughout the respiratory tract. This is when someone is at the most infectious, as millions of newly formed virus particles can be transmitted to someone else. through coughing, sneezing, or by contaminating various surfaces. One of the most important ways our immune system tries to fight the virus is by producing antibodies that are specific to the virus. Antibodies bind to proteins found on the surface of virus particles. By doing this, antibodies can prevent the viruses from infecting epithelial cells that line the respiratory tract. Antibodies are said to neutralize the virus. If someone mounts a strong antibody response to the virus, then these antibodies can be detected in the bloodstream. It usually takes between 14 and 28 days before a productive antibody response is mounted by our immune system. However, it is still not entirely clear how long people remain immune to the virus once they have been infected, or how long antibodies remain in the circulation. We know from studies of other viruses such as influenza virus that often antibodies can provide long-term protection as they persist in our bloodstream for a lengthy period of time after we have recovered from the initial infection. How long these antibodies persist and how well they provide long-term immunity to SARS-CoV-2 is currently not known. This has important implications when interpreting antibody tests. Antibody tests only detect the presence of antibodies to the coronavirus. They cannot determine whether a person is actually immune to the virus. More sophisticated tests and more detailed immunological analysis would need to be performed in the laboratory in order to determine whether someone is actually immune to the virus. How do antibody tests actually work? As most antibodies are most easily detected in the blood, then a sample of blood is required from a person. Although some antibody tests only require a droplet of blood, more accurate laboratory-based antibody tests normally require a larger amount. The sample of blood will contain lots of different proteins, although the large proportion of these proteins will actually be antibodies. Most antibody tests are based on a test known as an enzyme-linked immunosorbent assay, or ELISA. ELISAs rely on knowing the viral protein or proteins to which antibodies are produced against. In the case of SARS-CoV-2, this is normally the spike protein, which is found on the surface of viral particles. Any molecule that an antibody is able to bind to is known as the antigen. Assay plates are first coated with the antigen, in this case the SARS-CoV-2 spike protein, and the antigen determines the specificity of the test, as only antibodies specific to the antigen, and hence the virus, will bind. The biological sample, potentially containing specific antibodies, is then introduced to the ELISA plate. Only those antibodies specific to the viral antigen will then bind to the pre-coated ELISA. Normally, a secondary antibody, otherwise known as a reporter antibody, that is conjugated to a fluorescent or chemical probe, is required to provide a detectable and measurable signal. The signal is then detected by an instrument that can measure the strength of the signal. The signal's strength is directly proportional to the amount of original antibody present in the biological sample. If no antibodies are present, then nothing binds to the antigen. The test is therefore interpreted as negative. If antibodies are present, then they bind to the antigen and produce a signal that can be detected. The test is therefore interpreted as positive. Many such tests are currently being developed. However, any ELISA test needs to be highly specific, highly reproducible, and reliable. So let's just summarize. PCR tests detect viral nucleic acid, or the genome of the SARS-CoV-2 virus, and PCR tests determine whether the virus is present or not. PCR tests do not detect antigen. Antibody tests, on the other hand, detect whether our immune system has made antibodies to the virus. Antibody tests therefore indicate whether someone has previously been exposed to the virus. It's important to note that antibody tests alone do not determine whether someone is immune to the virus. More sophisticated immunological analysis is required to determine immunity.